Hello friends, welcome back to Multi-Question World. Today we are discussing important discoveries and inventions of microbiology. Anthony Phillips van Leeuwenhoek discovered single lens microscope. Anton von Leeuwenhoek discovered single lens microscope and named the microorganisms as little animalcules. Anton von Leeuwenhoek named microorganisms as little animalcules and he also discovered single lens microscope. Next is Edward Jenner. Edward Jenner developed the first vaccine in the world, that is the smallpox vaccine, by using cowpox virus. Edward Jenner developed first vaccine, that is smallpox vaccine, and this smallpox vaccine is developed by using cowpox virus. Next one, Joseph Lister. Joseph Lister is considered as the father of antiseptic surgery. He introduced the use of carbolic acid during surgery. Father of antiseptic surgery is Joseph Lister and he introduced the use of carbolic acid during surgery. Okay. Then Hans Christian Gram. Hans Christian Gram developed gram stain in 1884. The year is also important. Next one, Paul Ehrlich. Paul Ehrlich introduced the concept of chemotherapy. Chemotherapy is introduced by Paul Ehrlich and he also discovered salvarsan for the treatment of syphilis. Salvarsan for the treatment of syphilis. Next one is Ernest Ruska. Ernest Ruska invented the electron microscope in 1931. Electron microscope is invented by Ernest Ruska. Next one is Alexander Fleming. Alexander Fleming discovered antibiotic penicillin in 1928. Antibiotic penicillin was discovered by Alexander Fleming in 1928. Next one, Elie Metchnikoff. Elie Metchnikoff described phagocytosis and coined the term phagocytes. Phagocytosis is described by Elie Metchnikoff and also the term phagocytes is also by Elie Metchnikoff. Next is Kleinberger. Kleinberger described the existence of L forms of bacteria. Kleinberger described the existence of L forms of bacteria. Next is Barbara McClintock. Barbara McClintock described transposons. Transposons are jumping genes. So, jumping genes or transposons are described by Barbara McClintock. Next one is Walter Gilbert and Frederick Sanger. They developed methods for DNA sequencing in 1977. Walter Gilbert and Frederick Sanger in 1977. Next one is Carey B. Mullis. Carey B. Mullis developed PCR or polymerase chain reaction in 1983. Year is important for PCR also. Let us see some questions from this topic. Question number 1. Who discovered the single lens microscope? Edward Jenner, Anton von Leeuwenhoek, Joseph Lister, Ernest Ruska. Right answer is Anthony Phillips van Leeuwenhoek. Option B. Question number 2. Anthony von Leeuwenhoek called microorganisms as option A, tiny cells, little animals, little animalcules or germs. The right answer is little animalcules. Okay. Not little animals, it is little animalcules. Next. Question number 3. Who developed the first vaccine in the world? Alexander Fleming, Edward Jenner, Paul Ehrlich, Joseph Lister. Right answer is Edward Jenner. Next one. Edward Jenner developed the vaccine against cholera, tuberculosis, smallpox, rabies. Right answer, smallpox. Question number 5. Edward Jenner prepared the smallpox vaccine using measles virus, cowpox virus, polio virus, influenza virus. Right answer is cowpox virus. Next question. Who is known as the father of antiseptic surgery? Joseph Lister, Hans Christian Gram, Paul Ehrlich, Ernest Röska. Right answer is Joseph Lister. Question number 7. Joseph Lister introduced the use of alcohol, formalin, carbolic acid, iodine. Right answer, carbolic acid. Next is, who developed gram stain? It is Hans Christian Gram in 1884. Next question. Gram stain was developed in 1884, 1928, 1931, 1983. We have already discussed the answer. Right answer is 1884. Next one. Who introduced the concept of chemotherapy? Edward Jenner, Paul Ehrlich, Walter Gilbert, and Barbara McClintock. Right answer, Paul Ehrlich. Next one. Salvarsan was discovered by Alexander Fleming, Paul Ehrlich, Joseph Lister, Carrie B. Mullis. Right answer, Paul Ehrlich. Next question. Salvarsan was used to treat malaria, tuberculosis, syphilis, cholera. Salvarsan was used to treat syphilis. Next question number 13. Who invented the electron microscope? Ernest Röska, Leeuwenhoek, Fleming, Jenner. Right answer, Ernest Röska. Question number 14. The electron microscope was invented in. We have discussed already the answers, so you have to comment it in the comment box. Okay. Next one. Who discovered penicillin? Alexander Fleming. Penicillin was discovered by Alexander Fleming. Question number 16. Penicillin was discovered in 1884, 1928, 1931, 1983. This also you have to answer. Next question. Who described phagocytosis? Hans Christian Gram, Ellie McIntosh, Ernest Röska, Barbara McClintock. Right answer is Ellie McIntosh. Next question. The term phagocytes was coined by Ellie McIntosh. Next question. Kleinberger described the existence of dash. Viruses, L forms of bacteria, fungi, protozoa. Right answer, L forms of bacteria. Question number 20. Barbara McClintock described PCR, gram stain, transposons, penicillin. Right answer, transposons. Next one. Transposons are also known as silent genes, jumping genes, dominant genes, marker genes. Jumping genes. Next one. Who developed methods for DNA sequencing? It is Carrie B. Mullis and Paul Ehrlich, Walter Gilbert and Frederick Sanger, Jenner and Lister, Fleming and Gram. Right answer is Walter Gilbert and Frederick Sanger. Walter Gilbert and Frederick Sanger. Question number 23. DNA sequencing methods were developed in 1931, 1977, 1983, 1928. 1977. Next one. Who developed polymerase chain reaction or PCR? Right answer is Carrie B. Mullis. Question number 25. PCR technique was developed in 1977, 1983, 1931, 1884. This also you have to comment in the comment box. I hope this video is helpful for you. If the video is helpful for you, please share to your friends. And if you are watching without subscribing, please do subscribe the channel. Thank you.